Hello, and welcome to our quick lecture on history of microbiology. So, microbiology started actually back in around 300 AD when Hippocrates and his friends, his other fellow philosophers, came up with the concept of what's known as miasma. The miasma theory is that the miasma theory is basically that disease came from what they refer to as humors. And those humors were foul-smelling matter, so rotting meat, rotting vegetation, anything that had a foul smell, stagnant water, putrid water, sewage, those sorts of things. That disease came from those smells, those odors or humors that were coming off of those things. And we might say today they weren't that far off. However, they were affirming it to the actual smell itself as opposed to anything that may have been growing in that foul-smelling matter. So it wasn't until the 1500s, a gentleman, an Italian scientist, came up with the idea of the germ theory, and he believed, Fracastoro, he actually believed that there may be something in the actual matter itself, not just the humors, that was causing disease. So that was kind of considered the first maybe hints to what's known today as the germ theory. Later on, around the 1670s, another Italian scientist came up with the concept of germ theory. He's really kind of credited with disproving what's known as spontaneous generation, and that is Francesco Redi. We will take a look at his experiment in the next slide. And in this timeline, we have, of course, Hooke and von Leeuwenhoek, who both are credited with cell theory. We have Semmelweis. Semmelweis is thought of as the godfather of mothers because he came up with the idea of aseptic technique. Then we have John Snow, nope, not that John Snow, but an older John Snow, who demonstrated cholera bacteria and was one of the founders of the field of epidemiology. He mapped cholera around the city of London and discovered that cholera was coming from a single public well in London. And so he's credited with being the father of modern day epidemiology. Of course, Louis Pasteur, everybody's kind of knows about because of pasteurization in milk, but he also discovered the pathway of fermentation. He gave us our modern day understanding of fermentation. And his biggest contribution is he totally disproved spontaneous generation using an experiment called the S-flask experiment. Then we have a Lister. Of course, Lister is what to modern day Listerine is named after by Johnson & Johnson. Lister started using carbolic acid as a disinfectant, and that idea was picked up by Robert Johnson, one of the founders of Johnson & Johnson, and he sold a modified form of carbolic acid and ethanol to the public and health physicians and healthcare people, and he called it Listerine. And then we have Robert Koch. Robert Koch and his workers were able to determine the actual causative agents or bacteria that caused specific infections. And he created what's now today known as Koch's postulates. And Koch's postulates is used even at the CDC and other areas. We use the modified versions of Koch's postulates to determine the causative agent of a particular disease. So Francesco Redi, 1668, was the first one to record any kind of experiment that was used to disprove the idea of spontaneous generation. So in spontaneous generation, the idea is that life came from a vital force. And spontaneous generation had a religious background. It was thought as a branch off of miasma from Hippocrates and his fellow philosophers. And there were a lot of different characteristics of spontaneous generation that came together. But what spontaneous generation really was saying was that life came from non-living things, and life was a gift from God. Life was a vital force that was found in living things, but it wasn't living in and of itself. So Francesco Redi said, no, I don't believe that. I think that life is coming from life, and I think that we need to take a look at that. So he took pieces of meat, he put it in 3 different glass jars. Inside of those glass jars, he left one of them unsealed so that flies could come into contact with it. He sealed another one so flies could not come into contact. And then he kind of semi-sealed the third one by just putting some gauze over the top of it. And what happened is, of course, the one that was exposed that flies could get to, maggots would show up in the raw meat. In the flask that was sealed, completely sealed, and flies could not get into contact with that meat, there were no maggots found in that meat. In the third flask, when he put gauze over it, flies were attracted to it because they were attracted to that foul smell, that miasma type of smell. However, the flies could not get to the meat. So they laid eggs on the gauze up at the top, and so the maggots formed on the gauze. And what Reddy was able to show was that only where the flies were landing and laying eggs is where living things would show up. And of course, fly eggs are almost microscopic. They didn't understand that they were eggs at the time. And what Reddy was saying is life is coming from life. So he was addressing germ theory, right? That these maggots were coming from these, and also that life is coming from life. That's the idea of biogenesis. Then came along Needham and Spallanzani. Needham and Spallanzani were on this race to disprove or prove spontaneous generation. And Needham actually was attacking Reddy's work on spontaneous generation. And he boiled, he would take hay and chicken parts and boil them in water. And if he did not boil them, the water that the hay and chicken parts would soak in would get cloudy and go bad because microorganisms were growing in it. But if you boiled it for a short period of time, it would stay clear. But what would happen is a couple of days later, even if it was sealed, it would start to go bad. So he was saying spontaneous generation was true. He said it was an actual fact. But Spallanzani did the exact same experiment, but Spallanzani heated his broth longer to a higher temperature, and he sealed with something different. And in doing so, he kept any microorganisms out, and if it was unsealed, then it would grow bacteria. If it was kept sealed, then it would not grow any bacteria. Now remember, Needham had sealed his and still got broth growing. So what was going on? What was Spallanzani doing that Needham wasn't? Well, he was boiling it much longer. Why is this important? Well, because when Louis Pasteur created his S-flask experiment, another guy by the name of John Tyndall took a look at what Needham and Spallanzani did and figured a few things out. So we'll get to him in a minute. So Louis Pasteur really is credited with the germ theory of disease and with the idea of biogenesis, that life comes from life. And what Pasteur did with his S-flask experiment was just elegant and incredibly simple and just so well done. And it is considered one of the most important experiments in microbiology even to this day. So what he did, and the reason it's called an S-flask, is because he took a regular round bottom flask, filled it with a broth, some kind of nutrient broth, and then he took the glass part, the neck of the flask, and he melted it and elongated it into this big long S shape like that. So that when he boiled all of this broth in here, right, he filled it with broth. Once it was boiled, he would bring it to a rolling boil so that steam would run down through the flask and out of the mouth of the flask. And so this was all steam. Now, as long as he left this flask sitting upright, the broth would remain clear and there would be no bacteria present. But if he did this with a second flask and he broke off that S neck so that now the open part of the flask was directly exposed to the air could drop in and bacteria that were present in the air would begin growing. And so the bacteria would now be present and the broth would go bad. In the third trial, he took another S flask, he boiled it, and he then tilted it. And when he tilted the flask, the broth inside of the flask came into contact with this part of the S curve. He tilted it back, left it standing upright, and now bacteria began growing and bacteria were present in here. And what Pasteur was trying to show was that all three of these flasks were left unsealed, meaning this end down here, this end right here was open to the air. Air could enter into the flask and move around and come into contact with the broth. However, any microorganisms or particulate matter that may be found in air would get trapped right here in this curve of the flask because they are heavier than air and do not have the ability to move upwards. So they could not move up the flask and get into the broth. So air itself could contact the broth, but microorganisms in air would get trapped here. This is why if he tilted the flask and brought it into contact with this joint right here, this turn, then bacteria would be present a few days later. Now, Tyndall, just a couple years after Pasteur disproved spontaneous generation using his S flask, Tyndall wanted to provide more supporting evidence because there was still controversy about this. So he showed that the hay used in Needham's experiment contained heat-resistant bacteria. It was the same year that he discovered heat-resistant bacteria. Another guy by the name of Cohn discovered what we know in bacteria to be endospores. And endospores are heat-resistant forms of bacteria. So these guys made their discovery in the same year. That's quite serendipitous. And the endospore formation is what was showing up in Needham's experiment. Endospore forming bacteria are found in soil, and Needham was using hay. Hay is a grass. So when he was chopping the hay, there were endospores present on the hay, and even though he may have boiled and killed non-spore forming bacteria, the spore forming bacteria required a longer boiling time. So that is why Spallanzani's experiment did not show growth while Needham's did. So in addition to looking at some of these scientists in history, we need to compare our three theories here. So first we start out with spontaneous generation based on that miasma theory of humors, and there is a religious background to the idea of spontaneous generation, such as bad behavior, committing sin, punishment from God. And in spontaneous generation, it was believed that life came from a vital force and could arise from non-living matter. Then along came the cell theory, and the cell theory is that cells come from cells or pre-existing cells. And it is the basis for biogenesis because Pasteur showed in cell theory, not only do cells come from living cells, but that those cells must be living as well. So cells come from cells, but life comes from life. The original very first cell theory was introduced by two scientists by the name of Schwann and Schleiden, but they are the full development and introduction of cell theory is credited with Robert Hooke and Anton von Leeuwenhoek. Then finally we formed the germ theory, which is kind of moving along the same lines as the cell theory. And in the germ theory, which was proven by or developed by Louis Pasteur, as well as Tyndall, and you could even say perhaps Needham and Spallanzani, is that organisms not visible to the naked eye are able to cause disease. And that disease has a manifestation that is physical, not necessarily spiritual or religious. Of course, the original idea of germ theory is now credited to Francesco Redi, that Italian scientist who did the first experiment with the flies and raw meat, but the germ theory was made significant by Pasteur and his S-flask experiment. So here, once we had Pasteur entering into the picture here, he kind of opened up the door to what's now known as the golden age of microbiology. This is about a 60, 70-year period in which just the field of microbiology exploded. We will talk about many of these scientists throughout the semester, but those that are really important, of course, you've got Louis Pasteur with several discoveries, Koch with his germ theory of disease. He's the one who actually wrote out germ theory. And he also, you'll see him in here several times. He learned how to grow bacteria in pure cultures, and he found the causative agents for many different diseases and microorganisms. We have Nesser, who discovered Neisseria. We talked about Lister with aseptic surgery and Listerine. Another one that's really important, we have Finlay here. Finlay was from Cuba, and Finlay found that the Aedes aegypti mosquito was the vector for yellow fever. This was really important in the 1800s in Central and South America. We also have, where is he down here? Ehrlich here with the treatment for syphilis, Chagas for Trypanosoma cruzi, which causes Chagas disease, also in Central and South America and parts of southern United States. And Ehrlich here with the theory of immunity. Not that any of these other contributions are any less significant. These are just some of the more famous scientists and their discoveries. But I do want to talk about some scientists who are not so famous but have made some really major contributions to the world of microbiology. One is called, I believe I'm saying this correctly, Wansimus. Wansimus was a slave in Massachusetts in the 1700s. He convinced physicians in Massachusetts, in Boston actually, to mass inoculate people against smallpox. There was a small smallpox outbreak that was beginning in the city of Boston, and as a child in Africa, he had been inoculated against smallpox in a process known as variolation. Now, this is in the early 1700s. Jenner did not discover smallpox and cowpox and actually come up with the idea of vaccination until 1796. So this man here actually introduced the idea, the concept of vaccination based on something he had learned as a child in Africa before being brought to the United States. He was a slave at that time. And so he was able to allow Boston to avoid a smallpox outbreak, a smallpox epidemic. Harold Amis over here from 1918 to 2003 was a Harvard graduate who was key or instrumental in the development of modern day recombinant DNA technology. He discovered the ability to use RNA as a tool within microorganisms to mass produce genetic products. And his work is what allowed us to first introduce in the early 1980s. He led the charge to produce insulin using bacteria. So he took the human gene for insulin, introduced it into E. coli bacteria, and found that E. coli bacteria could produce human-specific insulin. In doing this, they were then able to introduce this insulin onto the public market, and the insulin that is used today to treat diabetics is still based on the recombinant DNA technology using RNA that Harold Amis came up with in the 1970s and 80s. Finally, Dale Emeagwali, who is still alive today. She is a graduate advisor at Excelsior College in Baltimore, Maryland, and she is the one who led to the modern worldwide or international understanding that microorganisms, bacteria in particular, are complex, even though they are prokaryotic. And she did this through discovering an enzyme that's a very complex enzyme produced in a bacteria known as Streptomyces. She may actually be more famous for her work in oncology. She is both a microbiologist and oncologist, and she discovered the role of a mutant protein called a RAS protein that is a major component of most forms of cancer. And she created and discovered the function of this protein using a technique she developed with colleagues called short interference RNA, which is now the #1 or standard method used to determine the function of proteins within cells. Just a couple others that are important. Here's Oswaldo Cruz, who used work from Carlos Finlay that we talked about a little earlier from Cuba. Oswaldo Cruz was in Brazil, and he eradicated yellow fever in both Rio de Janeiro and Belém de Pereira in Brazil. I hope I pronounced that correctly. He also introduced into Brazil and other parts of South America the concept of preventative medicine. Carlos Finlay, we talked about in Cuba, discovered that Aedes mosquito was the vector of yellow fever, and in doing so, he was able to introduce mosquito eradication efforts throughout parts of Cuba, Central America, and parts of South America. Then we have Mary Hunt. Mary Hunt I like because she was nicknamed Moldy Mary. Alexander Fleming actually is the one who discovered penicillin. Penicillin was the first antibiotic ever discovered, and it was extracted from a mold known as Penicillium, Penicillium notatum. The problem was that we wanted to make lots of this antibiotic. We needed lots of penicillin. We knew that it was effective, but we didn't have enough of it. And they wanted to mass produce it because at the time that Mary Hunt was in, this was done in Peoria, Illinois. Once Mary Hunt was in Illinois, this was just during World War II. So they wanted to have this antibiotic available for soldiers that were injured in World War II to prevent infection. And Mary Hunt is the one who finally did the research and discovered the final strain of penicillium that to this day is still used to mass produce the antibiotic penicillin. So she was nicknamed Moldy Mary. By the way, she discovered that mold on a cantaloupe. And then we have June Almeida, a virologist, and I saved her for last because she was amazing. She's from England, and in 1966, using an electron microscopy technique that she founded and developed herself, she discovered the first coronavirus, which is the predecessor to COVID-19. And she actually was the first person to ever, using an electron microscopy, allow the visualization of any virus ever. So it was a woman who first discovered the structure of a virus and allowed and was able to develop a microscopy method so that we could see viruses. It just so happened that some of the viruses that she discovered, one of them was the first coronavirus, same family as the COVID-19 and the SARS and MERS virus in the latest pandemics. So these are just some additional scientists that had some major contributions to the world of microbiology. And I hope you understand that although they have some really major contributions, they do have others as well, as there are hundreds of other scientists out there that their work also contributed to what these guys have done and have built upon. So I hope you enjoyed our lecture on a little bit of history and microbiology, and I look forward to moving into scientific theory next.